with regard to phylogenetic tree construction, there are certain basic idea regarding that. Mainly we are using phylogenetic tree construction to find out the evolutionary relationship between two species or a group of species or a group of genera. That is the primary aim of any kind of phylogenetic analysis, and usually we are obtaining this phylogenetic similarity by constructing an evolutionary tree or a phylogenetic tree. So that is the primary aim of a phylogenetic analysis. Now we will be using different types of methods for that, but there are certain basic principles or basic assumptions. For example, if you take any organism, it has a nucleus, it has a chromosome, and chromosomes containing genes, and genes are made up of DNA. So DNA, RNA and proteins are present in almost every organism. There are two fundamental principles that is shown here. In general, closely related organisms have high degree of similarity in the molecular structure of this substance. That is, if you take the DNA or RNA of two species, for example, you take a lion and tiger, both are coming under the cat family, there will be some similarity between the DNA of a lion and DNA of a tiger or DNA of a leopard, because all those belonging to the same group called as cat family. But if you take the DNA of the lion and an elephant, we may not be able to find so much similarity in their DNA. So closer the animals, higher the similarity. So that point is that while molecules of organism distantly related show a pattern of dissimilarity. So if you compare the DNA of a lion and the DNA of a tiger, DNA of a leopard, we will be seeing a lot of similarity between them. But if you compare the DNA of a lion with the DNA of a whale or DNA of an elephant or DNA of a deer, we will be seeing more amount of dissimilarity or more amount of differences between them. So the principle is that more closer the organism, more will be the similarity. More different the organism or more distantly related the organisms, their DNA will be having higher dissimilarity. This is the one basic principle. Another is that conserved sequences such as mitochondrial DNA are expected to accumulate mutations over time, and assuming a constant rate of mutation provides a molecular clock for dating divergence. So while studying about cytology and also in genetics, you have studied about this extranuclear chromosome, especially the mitochondria you might have studied. In the mitochondria we have DNA, and one important peculiarity of this DNA is that they are very slow, they are highly resistant to any kind of mutation. So mutation or evolutionary change are very slowly taking place in this mitochondrial DNA, so that we call it as conserved. Conserved simply means exist without much change. That is the meaning of the word conserved. So DNA which will be present in the mitochondria are usually very conserved. That is, they remain like that for millions of years without much evolutionary change. So that is the idea of conserved DNA or conserved DNA sequences. So usually we are while taking the DNA fingerprinting, we are usually taking this mitochondrial DNA. Even if you are sending a specimen for that, we are actually taking a mitochondrial DNA CO1. So that is the meaning of the word conserved sequence, because they remain like that without any kind of change for a long time. Molecular phylogeny uses such data to build a relationship tree that shows the probable evolution of organisms. So by studying about those DNA which are present in the mitochondria of different organisms, we will be able to get how similar two organisms are there. That is why we are usually taking mitochondrial CO1 gene. It's a type of gene present in mitochondria, and this mitochondrial CO1 gene will be giving us a lot of idea regarding the evolutionary relationship between these two organisms. Now we come to the top term known as phylogenetic tree. It is basically an evolutionary tree or a tree-like branching diagram showing the inferred evolution. Inferred means guess. That is a meaning of the infer mean to guess something. We are showing the inferred evolutionary relationship among various biological species based upon the similarity and differences in their physical and genetic character. A long sentence is there. But here if you look at that, this is an evolutionary tree or a phylogenetic tree. What it is showing the inferred, what is assumed, because this is how we assume that living beings are connected with one another. So that is what we are showing in this phylogenetic tree of life. So inferred evolutionary relationship based upon similarity and differences, and this tree has been constructed based upon our knowledge of what is similar and what is dissimilar. That is the idea that is shown. Taxa joined together in the tree are implied to have descended from a common ancestor. For example, here all these organisms are commonly joined here. That means all these organisms have a common ancestor. That is why they have a common point here, or a common origin here, or a common root here. Here also you can find all these organisms have a common origin here. So that is here also you can. When they are all connected by a common node, that means they all arised or evolved from a common ancestor. That is the idea which is shown here. Now we are coming to another few more points in the phylogenetic tree. Tips of the tree represent group of descendant taxa of a species, and nodes on the tree represent their common ancestor. Here you can see that. Here this is a very simple evolutionary tree. Here you can see that this is the tip of the tree, and this is the common point, or this is the root of the tree. Now what is mentioned here is that tips of the tree represent descendant. Descendant means what is formed, what is evolved as a result of evolution. So descendant taxa, normally we are calling it as a species. So here you can say that this is one species and we call it as taxon A, and here there is another species, we call it as taxon B, and there is also one more species which we call as taxon B. So these are the tips of the tree. So here one species is there, here another species is there, here third species is there. And what is mentioned here, the tips of the tree represent the descendant taxa, and nodes on the tree represent the common ancestor of this descendant. Here you can see that there are certain common points. These are known as a nodes. Here also there is a common point. This is known as a node. So here in this diagram we are finding one node here, one node here. So what is the meaning of the word node? Nodes on the tree represent common ancestor. That means here there is a node. For the taxon species A and species B, or taxon A and taxon B, they used to have a common ancestor, and that ancestor is represented here. So the common ancestor of A and B. Here also we can find a node that will be the common ancestor of A, B and C. So that is the idea that we are getting from an evolutionary tree. An evolutionary tree will be showing what is known as a tips will be there, and nodes will be the tips will be representing species evolved, and nodes will be showing their common ancestor. Now again few more points regarding that. The two descendants that split from the same node are called sister groups. Here you can see that here taxon A and taxon B, they are known as sister groups. Because why they are known as sister groups? Because both taxon A and taxon B have evolved or diverged from this common ancestor. So when two species are diverging or evolving from one common ancestor, we are calling it as a sister group. Now what is shown here next? Many phylogenetic tree also include an outgroup, a taxon outside the group of interest. So outgroup stems from the base of the tree. Now here you can see that here this taxon A and taxon B, we are normally calling it a sister group. Now what is the case of taxon C? Taxon C is not directly evolved from this common ancestor. Instead, it has evolved from another earlier common ancestor belong common to common A, B and C. So it is from that earlier ancestor this taxon C is actually formed. So we call the taxon C as an outgroup because taxon A and taxon B are directly evolved from one common ancestor. That is not the case for taxon C. So taxon C is somewhat more distantly related with the taxon B and taxon A. So we call taxon as C as an outgroup. So there are two terms here. One is sister groups. If they are evolved from same common ancestor, we call it as sister group. But if it is evolved from a previously common earlier ancestor, we call it as by the name outgroup. So here you can see taxon C is an outgroup. See that example, human and mouse, they have a common ancestor. Then, but if you take the fly, they do not have a common ancestor, but still they also have an ancestor similar somewhat many generations or many millions of years back. So that is what, see, fly is an outgroup, mouse and human is shown as a sister group here. So that is how that is all present. Now one more term, that is a clade. Clade is a group of organisms that includes an ancestor and all the descendants of that ancestor. For example, you can think clade as a branch on the tree of life. Or here you can see that this here there is a common ancestor and there is one species A and another species B is here. Now this entire thing, taxon A, taxon B and this common ancestor, they are combined together known by the name a clade. So clade is defined as a group of organisms that includes an ancestor and all their descendants. So here taxon A, taxon B and this common ancestor will be considered as a clade. So number of terms I have introduced. One is sister group. If they are originating from one common ancestor, we call it as sister group. Then there is outgroup. If they are somewhat distantly related, not directly evolved from the common ancestor, we call it as outgroup. And in these tips of the taxa showing descendant, nodes will be showing the common ancestor. This entire thing, concentric tips and the common ancestor are called as a clade. So these are the points which are mentioned in this part of the slide. Now what are the applications of phylogenetic tree? Sometimes you can expect for as a short answer or a 4-mark question. So understand the evolutionaries. That is the primary importance of any evolutionary tree or a phylogenetic tree. Now in the case of pathogen means disease causing microorganism. We will be, they are very small. So by doing a DNA analysis, we will, because all bacteria, microorganisms, they do not show much morphological differences. They all look very similar. So by only doing a DNA study, we will be able to understand their evolutionary differences or evolutionary similarities, etc. Then assist in the epidemiology of infectious disease or genetic defect. For example, when corona, it was a new virus, COVID, it was a new virus, but doctors were very quick to point out that the mode of reproduction and mode of infection and mode of incubation and how the symptoms that will be producing and what are the organs that will be infected, all will be similar to our common cold virus. So that is their prediction. They could predict that because they have studied the common cold virus for a long time. So the working of the new virus is also somewhat similar to the common cold virus. This knowledge, this information is what is mentioned in the epidemiology. Then aid in the prediction of function of novel genes. That is another peculiarity of the phylogenetic tree. Sometimes we may be able to find certain new genes, but by studying about the similar genes, similarity of this new gene with other gene, we will be able to predict what will be the function of this new gene which has been discovered. That is another advantage. Biodiversity study, this is very lot of applications, that is why we are also using it, because by doing a biodiversity DNA, now because many of them have sent specimens for molecular analysis, they all got DNA sequence. Now by simply going to the NCBA GenBank, we are able to find out this species of that I have collected is belonging to this particular organism. 99% it will be similar to that. So that way that is a very important tool for biodiversity analysis. Then understanding the microbial ecology. If you get a new bacteria or a new virus, we do not know how it is functioning, how it is reproducing, how it is feeding, how it is surviving in the environment. But if you find that it has some similarity with some bacteria or some fungi that we already know, then we will be able to predict the function or mode of living of this new bacteria also. Then it is involved in multiple sequence alignment. Then with regard to conserved sequence also, I told you conserved sequence are those DNA which remain in the organism, especially in our mitochondria, for a long time without much evolutionary change. That also it is possible to find out using phylogenetic tree. So what are the steps in a phylogenetic analysis? We first go for the selection of organism. Then we'll be choosing which molecular marker or which gene has to be analyzed. And once we know that this is for example, most of the time we are sending the DNA of CO1 gene present in mitochondria. Another gene that we are sending is cysteine RNA. So then we also send 28 RNA. So these are the common type of genes or markers which we are using, and we will be amplifying, means increasing the, if there is only 10 mole gene, we will be weighting it first into 1000 or 10,000 gene. That process is known as amplifying. Then we extract DNA from that, then we will be aligning it, and finally we are conducting a phylogenetic analysis, then go for tree. For example, last time when I was doing with the mega software, I showed you how this multiple sequence alignment are doing. Now your student, now some your classmates are also doing this multiple sequence alignment. Once they align the multiple sequence alignment, they use that data for a phylogenetic tree, because nowadays with the software it is very easy. Once you try with a few times, you will be able to get a evolutionary tree. So this is how normally an evolutionary tree are constructed. Different type of software is that. So you know what is a cluster W. Last time we have seen that cluster W, how we are doing that. So then using this mega software, number of softwares are the most easier and most common software is Mega X, because it is a free software which we can download. Otherwise, free software always have its own limitation. But paid software will be incorporating newer information, newer data also. So one common phylogenetic packages are POP, PAML, Phylip. All these are phylogenetic tree building software. So there are number of methods are there, and all these methods here also you can see Panther, Peapod, PFAM, TreeFam, PhyloFact. So lot of other phylogenetic tree building software are there, and some are based on distance based method. I am not going into the details of that. And some like a UPGMA, this is another type of approach in phylogenetic tree construction is used. Then we have neighbor joining. While you are going for phylogenetic tree construction, you might come across these kinds of terms known as neighbor joining. Then maximum parsimony. Maximum parsimony is actually based on one principle. That principle is on the principle of Occam razor. That states that the simpler hypotheses are preferred to complex one. That is, when we are building a tree construction, that principle is also combined with the principle of parsimony. That is saying that phylogenetic tree with least number of evolutionary steps are often preferred by nature. For example, we are having two trees will be there. According to the principle of evolutionary parsimony or maximum parsimony, this is one tree we have constructed, this is another tree we have constructed. But this principle of maximum parsimony states that which tree is the most preferred tree? The tree which has least number of changes. Here there will be two changes, here there are only one change. So nature will be always preferring this tree. That principle is known as principle of parsimony. Parsimony means in Malayalam we call pisik. So nature also have that approach. So parsimony means nature prefer to spend only least amount of change. So that is why we call this tree as the best tree, because here nature has to expense more changes. Nature has to afford more changes. The nature do not prefer. So this is the principle of parsimony or principle of Occam-Glaser. Then regarding the phylip is a phylogeny interference package. The primary purpose of phylip is that phylip is mainly used as a phylogenetic tree construction. Now we don't use phylip much because there are more common, more simple packages are there, especially mega-X. So mega-X is one of the most common, widely used tree construction for phylogenetic tree.